Hey everyone, welcome to week 2 of conservation biology. So I do have a couple reminders for this week. Due this Friday, January 15th at 5 p.m., you're going to have some similar things to last week. You'll have 2 discussion posts. You'll have quiz 3, which will be on genetic diversity, which is this lecture. You'll have quiz 4, which is going to be on extinctions and ecosystem degradation. That's actually going to be 2 lectures, but they're relatively short, so I just combined them together so you can just do one quiz. And then you're going to have a quiz on your week 2 readings, which are in the book Where the Wild Things Were. You're also going to have your second species worksheet due, labs 3 and 4, which are already uploaded on my courses. And you're going to have 2 things that are a little bit different this week. You're going to have your video or literature review. That's all uploaded on my courses. There's a folder for it in the content tab. And there's the option of either watching the video on there, which is a documentary, or reading the 3 papers that are on there. And then for whatever you choose, you'll do a short review. And then you'll also have your research paper preliminary references due this week. So a little bit more about your research paper, just as a reminder. Everyone turned in their topics this past week, and they looked really good from what I've seen. I haven't finished going through them as of this point, but I will send them out to everyone before the end of the weekend. I'll send out my notes, letting you know if it's good to go or if I have anything to say about it. So this week, your task is to do some literature searches. So I would suggest using Google Scholar. They're going to have a ton of peer-reviewed articles and journals for you that you can look through, and find at least 5 references that you think are going to help you with your paper. It's okay if you find these 5 references and then when you're writing your paper later on, you don't use them all. It's not saying that you absolutely have to use these 5 references in your paper. You can use different references later on. But you do want to find 5 that you think will work, and you want to make sure that you're seeing enough literature out there so that you'll be able to write your paper. Remember that your paper should, at minimum, include the background on your topic, so you want to start broad and then narrow down to what your specific topic is. You want an overview of the current issues with your topic, so anything new that's happening, kind of like a timeline of how things have been going with your issue. You want examples of any species or habitats that have been impacted. You want to talk about any legislature that has been implemented that addresses your topic. You want to come up with some things that can still be done to help. So this can be something you found in an article, or it could even be something that maybe you came up with on your own. And then you want to draw some conclusions. So you want to kind of wrap it all up. So look for papers that include some of that information. That's going to help you. And then make sure your paper is stressing the importance of how humans are contributing to your issue. Because really this whole paper is supposed to be about a human impact, right? So you want to make sure that's being stressed. And then when you're writing your paper, each of these sections can have multiple components. You don't have to just have those 6 things, right? You can have subsections beneath them. You can have whole other sections as well. If you come across anything in your literature search that you think is interesting, add it to your paper. If it's about your topic, it can be there. So don't feel limited by those 6 things. That's just the minimum of what you need to include. You can include anything else you want to include. Okay, so also in next week, so due, let's see, what's that date? Due on the 22nd of January is going to be your midterm. For your midterm, you have a choice of 4 out of 5 essay style questions, and then an extra choice of a mathematical or an analytical question. I'm going to have the midterm study guide uploaded to my courses today, so that's Saturday. You'll have that up there by the end of today. The exam is open book, and you do have a time limit though, so you have 2.5 hours. So even though it's open book, you want to make sure you go through the study guide and really maybe write down the answers to some of the topics, like write down descriptions of some of the topics. And that way you have it prepared and you're able to do the essays much more quickly. It shouldn't take 2.5 hours anyway. I gave you a lot of time, but do make sure you prepare a little bit ahead of time and make sure you go through the study guide. The study guide has all of the topics that are fair game for the exam. You also have to do the mathematical or analytical questions. So I have topics for that, and I have a list of the mathematical type things that you have to be able to do. So make sure you look at that study guide before you do the quiz. You have a whole 2 weeks now until the quiz is due, until the the exam is due. So make sure you take a look at it. You can run through it this week and just write down all the answers, and then next week you'll be set to be able to take the exam. I promise it's not going to be too difficult. It's going to be big concepts. I'm not going to be asking you guys to define like 100 different terms. It's going to be big concepts, making sure you understand the overarching themes of the course. So finally to what we are doing today. So this lecture is on genetic diversity, which, if you'll remember, is the third level of biodiversity. And genetic diversity can refer to diversity among members of a population. It can refer to the genetic diversity within a species. It can even refer to the genetic diversity within one individual. So it can be looked at on multiple different levels. It's not all just about genetic diversity in one individual. Sometimes we see things that have different appearances and we assume it's a genetic difference, but it isn't always the case. So not all physical differences have to do with genetic variation. Sometimes variation is due to an environmental condition. So that's the case here with these hydrangea flowers. For this type of flower, the flower color depends on the pH in the soil. So if the soil is basic, then the flowers are pink, and if it's acidic, then the flowers are blue. It has nothing to do with genetic differences. So in fact, you can actually have one hydrangea plant, and if you change the pH of the soil that hydrangea plant is in, it's going to change the color of the flower. So one plant can go from pink to blue depending on what the pH is. However, some genetic differences have evolved due to environmental factors, but they are still genetic differences. So one example here is human skin color. Darker skin colors come from areas that are closer to the equator, and there's a scientific reason for that. It's because darker colors are protect you from the sun's rays. So they needed a darker skin tone in order to protect from those direct rays that you get in the center of the earth and by the equator. Whereas lighter skin colors originated near the poles. And you probably know by the poles, the sun's rays are not nearly as direct. And in those areas, they needed lighter skin so their skin could absorb more of the sun and get the necessary vitamin D that they needed. You'll also probably notice people who live in coastal regions have darker skin. And the reason for that is because if you live in a coastal region, you have more access to seafood, which is rich in vitamin D. So they were able to eat a lot of seafood and get their vitamin D, which means they didn't need that light skin to absorb more vitamin D, and they benefited from having the darker skin that protects them from the sun's rays. So even though skin color is determined by genetics, the genes were selected for a very long time ago by environmental factors. So when we look at genetic diversity at different levels in humans, we actually see that almost 88% of our diversity is seen between individuals. So it doesn't matter if they're from the same population or not. So for example, if we looked at the genetic diversity in Binghamton, New York, that would account for the majority of the genetic diversity we see across the world. We see the same levels of genetic diversity and the same type of diversity everywhere you look on Earth. It doesn't really matter where someone's from. Only about 9% of our variation comes from what continent we're from. So it's a very small amount that comes from a geographical area. So when you look at people all over the world, you need to realize they're all very genetically similar, no matter where you're from. So I'm just really quick going to go over a couple of terms that you should understand to have a little bit of a better understanding of the next few things we're going to talk about. So you're not going to be tested on this slide. This is just to make sure you are caught up with what we're going to be talking about in a minute. So genes are portions of your DNA or RNA that can code for a trait. They actually code for a protein, but through a lot of steps, it codes for a trait. So there's actually a section of DNA that's found on a chromosome, and many traits are coded for by multiple genes. So it's not necessarily just one gene that codes for a trait. Then we have alleles, which is a variant or alternative version of a gene, and that determines how a trait might present itself. So in diploid organisms like us, we have 2 sets of chromosomes, one from each parent, and there's 2 copies of each gene. So if the 2 copies are identical, so that would be like here, so we have 2 of the same letter, then that individual is homozygous at that gene locus. So homo meaning same, and zygote, same zygote. So that individual will be homozygous. But if the 2 copies that come from each parent are different, then they would be considered heterozygous. So big B, little b would be heterozygous. So I'm going to use an example of eye color. So there's a bunch of genes that code for eye color, but we just picked 2. So we have OAC2 and GEY. So OAC2 has 2 possible alleles. So it has big B for brown or little b for blue. So those are the 2 alleles that it has. GEY has 2 possible alleles as well. You have big G for green or little b again for blue. So now it's important to note that we have uppercase and lowercase letters here. The uppercase letters are the dominant alleles, so if they are ever present, they are going to be what is shown over the recessive allele. So here we have big B, little b, and even though there's a blue allele there, we're getting brown eyes because B is dominant. Okay, now for eye color, we're looking at multiple different genes. So we have OAC2 and GEY, and you can see there's actually different levels of dominance. So even though there's 2 big G's and 2 big B's, we still got brown. And that's because brown is the most dominant. It is more dominant over G, over green. So in order to get a different eye color other than brown, you cannot have any brown alleles in order to get a different eye color. So that's why brown is the most common eye color, because if there's even one brown allele, you have brown eyes. In order to get green, you have to have at least 1 big G and no browns. So big G and then 3 blues, or big G and 2 blues. That's how you get green. And then blue is recessive, which means that if there are any dominant alleles available or any dominant alleles present, then you are not going to get blue. So the only time you can get blue eyes is if you have all recessive alleles. So it doesn't really explain everything. So this doesn't explain why 2 parents with blue eyes can have a child with brown eyes, because theoretically they would only have blue alleles, right? So how would their child have brown eyes? It also doesn't explain how gray or hazel eyes arise. So that's where there's modifier genes that come in, and other genes that are associated with eye color, and also mutations. So all of those things can lead to variability in eye color. This is just a very basic description. There's a lot more to it, but you only need to know the basic type stuff right now. So we're not in a genetics class. We don't have to know a ton of genetics. So two more terms that you might want to understand are genotype and phenotype. So the genotype refers to the number of different genetic combinations of alleles are possible. So in the eye color example that we gave here, there would be 9 different genotypes. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So there's 9 possible combinations here. The phenotype is referring to the physical output that the genotype has. So even though brown has 6 different combinations, that's one phenotype. So that's one physical property that comes out of the 6 different genotypes. So there are only 3 phenotypes, but there's 9 genotypes here. And one final thing that I want to mention is allele frequency. And what that is it's just simply the frequency that a certain allele is present in a population. So how many individuals in the population have this particular allele. Allele frequency isn't stagnant, so it can change really rapidly. We're going to learn about a lot of ways that the frequency of alleles can change, but it's really just how many individuals have this. So let's say we had 9 individuals here, and each one just so happened to be a different genotype. So we had these 9 genotypes. If you're looking at the frequency of G, you would just add up all the G's, and it would be 1, 2, 3, 4, 5, 6, 7, 8, 9, and then you'd add up everything else, and it's 9 out of however many alleles there are. So what we're talking about here with changing allele frequency, different allele frequencies, is natural genetic variation. Natural genetic variation. And this can occur at multiple levels. So you can have it among higher taxa. You can have it among related species. You can have natural variation among populations of a species, among individuals of a population, and even within the individuals. So we mentioned homozygous or heterozygous. So each individual can be homozygous or heterozygous at a particular gene. And even with that, they could be homozygous for a different allele than another individual is. So, for example, this individual is homozygous for brown, but this one's homozygous for blue, and this one's homozygous for green. So there's all different possible combinations that you can have here. But the ultimate source of genetic variation is really mutations. They are what cause the majority of genetic variation. And the primary way that genetic variation is lost is through something we call genetic drift. And we're going to define all of this in a second. So mutations happen all the time. They are considered the change, addition, or deletion of a nucleotide base. So they're random, but certain factors can happen that make them more likely to occur. So external factors like radiation can cause mutations, or even internal factors like DNA replication can cause mutations. So the more you replicate DNA, the more likely there is for a mutation to occur. Some mutations have no effect at all, and they go unnoticed. Some of them have an effect, but they don't really harm or help the organism in any way that we know of, at least. And then some mutations are kind of detrimental to an organism and causes them to die, or even be ill, or never even possibly fully form. So some mutations can benefit the organism, can lead to evolution, which is great, right? Evolution's always, well, not always. Evolution is typically a good thing. But even then, not all beneficial mutations are passed on to future generations. However, if a trait is advantageous, and it helps the individual survive and reproduce, the genetic variation is more likely to be passed on to the next generation. And over time, as generations of individuals with the trait continue to reproduce, the advantageous trait becomes increasingly common in a population because it's being passed on more and more. And eventually that can make the population different than one of their ancestral populations. And sometimes they'll become even more different that they become considered a new species. So that's one of the ways that evolution happens. But not all mutations lead to evolution. Only hereditary mutations, which occur in egg or sperm cells and can be passed on to future generations, those are the only ones that potentially contribute to evolution. So traits that are given from your mother and father. Some mutations occur during a individual's lifetime. And it can occur in only some of the body's cells, and they're not hereditary. So natural selection can't play a role in them. Also, many genetic changes have no impact on the function of a gene or a protein, and they're not helpful or harmful, like I mentioned earlier. Some of them have no known impact. In addition to that, the environment in which a population of organisms lives is integral to the selection of traits. So differences introduced by mutations can help them survive in one setting, but not in another. So for example, a resistance to a certain bacteria is only advantageous if the bacteria is found in a particular location and it harms the individuals that live there. So if you don't have that bacteria, you're not gonna have a mutation that will be passed on because it's not helpful to you. So if we're thinking that only helpful ones pass on, so why do we have harmful traits like genetic diseases that persist in populations instead of just being removed by natural selection? And there's a few possible reasons for this. In many cases, the answer is not really clear, but for some conditions, such as like Huntington's disease, the signs and symptoms don't occur until after the person has children. So the mutation can be passed on even though it's harmful because you don't know about it until afterwards. For other harmful traits, a phenomenon called reduced penetrance, in which some individuals with a disease that have this mutation don't show any signs or symptoms of the condition, but they can also allow harmful genetic variations to be passed on to future generations. So they have no symptoms, but they have this harmful mutation. And for other conditions, which is really interesting, this is what happens with sickle cell disease. If you have one mutated copy of this gene, it's actually good. It's advantageous because it actually provides protection against malaria. But if you have two copies, that's when you get sickle cell disease. So having two copies of this gene is called the HBB gene in each cell results in sickle cell anemia, which is when your blood cells, they actually kind of change shape and they don't hold oxygen the way they're supposed to. So having one copy, you have resistance to malaria. If you have two copies, you have sickle cell anemia. So it gets passed on because it's actually advantageous to have one. So what I want to get into next is going to be genetic drift, but first it's important to understand that there's differences between genetic drift, natural selection, and speciation. So I'm going to describe natural selection and speciation first. Natural selection is a mechanism of evolution. It usually occurs when an environmental cue or a pressure leads to certain traits becoming more fit and allowing those traits to be passed on. So a really amazing example where we actually got to see natural selection occur at a relatively fast pace was with these peppered moths. So originally, these moths were white in color. So they looked like the guy up top. It was white, a little bit of black speckling. Black individuals, like this guy on the bottom, were really rare. And you can probably guess by looking at the picture as to why they were more rare. It's because this guy blends pretty well. This guy stands out like crazy on tree trunks. So they were able to be seen by predators and they were eaten. The white moths were better at camouflaging. So what happened with them is during the Industrial Revolution, scientists started noticing that the black individuals were becoming more and more common. And it was to the point where they actually ended up making up like 98% of some populations of these moths. So they were more common than the white species. They were almost the only moth around was the black individuals. And so you might be asking yourself, like, what caused this? So what happened is it turns out that in the Industrial Revolution, soot from the factories that were being built was covering all the trees and turning everything black. So now when the white moths sat on the black trees, they stood out right away to predators. And the black moths were able to camouflage, and therefore they were able to survive long enough to pass on their genes to the next generation. So because they were able to survive and breed and produce offspring, since they blended with the black trees, they became the dominant moth. Since the Industrial Revolution, we've actually gotten better. And, well, in some ways, we've gotten better with that type of pollution. So we have less soot in the environment and the trees are no longer black. But so now what's happening is the moths are actually reverting back to being predominantly white again. So you can see selective pressures changed their allele frequencies because this is a genetic difference, having either a brown color or a white color. But the selective pressure was able to change that. So it didn't cause a loss in genetic diversity the way that you're going to see genetic drift does. So that's a very important difference here is genetic drift. Well, with natural selection, it doesn't mean that the allele is completely gone. With genetic drift, a lot of the times you completely lose alleles. In this one, you're just changing the frequency, and it's due to selective pressure. So it's something that is not random, right? They're being selected for because they are better at some certain thing. So this example is actually a really great reason why genetic diversity is important, right? If these moths didn't have this diversity or their ability to change like different colors, then they most likely would have just died off when the trees turned black. If all of the moths were white and they just stood right out against the tree, they would have all been seen and they could have all been eaten. But because they had genetic diversity and they were able to change the allele frequency, then they were actually able to survive through this time and now again change back and survive on the lighter trees again. So speciation occurs when populations are reproductively isolated from one another. So when I say isolated, that can mean a few different things. You can have allopatric isolation, where there's a geographic barrier separating populations. So this is like what we discussed with the evolutionary significant units, where we had the physical barrier or the river that widened, and now the species are separated and they slowly change over time due to different types of natural selection and different types of genetic drift, which we will get to in a second. So over time, all the different pressures that are being put upon them are separate from one another. So they're able to mutate and reproduce with each other and eventually become a different species. Or at least a different population, different subspecies, anything like that. The other type of speciation is sympatric isolation. And this type of isolation happens to populations that are reproductively isolated, even though they live in the same area. So there's a lot of different ways that this can happen. They can have barriers that are considered prezygotic barriers, which are barriers that were decided before the egg was even fertilized. So these are genetic barriers. Some examples of these prezygotic barriers are behavioral isolation. So we mentioned in one of our lectures last week the eastern and western meadowlark. So here, the eastern and western meadowlark, they look very similar. Their ranges overlap, and they have very different songs. And it turns out that even though they look very similar, you would think, oh, maybe they can interbreed. They won't. They won't interbreed with each other. And the reason is they will only go for an individual who has the song of their species. So they have a barrier there. They can be found in the same place, but a behavioral preference keeps them from being able to mate. So another prezygotic barrier is timing. And this is called temporal isolation. So this is particularly an issue for plants. It's not really an issue for animals. But for an example, if a flower blooms at night and another flower blooms during the day, they are not going to be able to breed with each other. So it just depends on what their preference is for what time they are able to breed. Now this can happen over different seasons. It can happen over years. It can be a difference in months. It can be a difference in weeks, days, or even hours. So that's, night or day. So there can be two very related plant species that just happen to bloom at different times, and so they will never be able to mate. So they are different species. Then you have another possibility, which is their habitat preference. So even though two species may live together in an area, they might differ in where they prefer to live. So for example, two types of frogs. One can live in the water like a bullfrog, and the other one lives up in the trees like a gray tree frog. They live in completely different areas, so that isolates them based on their habitat preference. Then you have postzygotic isolation, or postzygotic barriers. So this is a type of barrier that occurs after the egg is fertilized. So this can occur if two individuals mate that are unable to produce fertile offspring. So like in mules. These individuals, they're isolated from one another now. So even though you can have them interbreeding like horses and donkeys, they don't produce fertile offspring. So reproductively and genetically, they are isolated from one another. So it doesn't have to be that they produce a non-fertile offspring. There are also species that can reproduce and produce offspring, but the offspring simply aren't healthy. So maybe they don't survive to maturity, or they're not even strong enough to fully mature in the womb or be born. Sometimes you can even have postzygotic isolation that can combine with sympatric isolation. So if populations are separated from one another for long enough and somehow eventually are reintroduced to each other, the postzygotic isolation, due to the genetic changes that each population went through, might keep them from being able to ever breed again or ever share their gene pool again. So the sympatric isolation can lead to postzygotic isolation. So finally, we are at genetic drift. And we're going to spend quite a while talking about genetic drift and types of genetic drift. So this is the primary way that genetic variation is completely lost. It's a sampling error in the transmission of alleles between generations. And a sampling error pretty much means how likely is it that we will have the same proportions of alleles from one generation to the next generation. So to put it differently, it's really the mechanism responsible for the random changes in how the gene pool is made up. And when genetic drift happens, the proportion of alleles changes. Sometimes certain alleles can be eliminated altogether. And what's important to note is that genetic drift is due to chance. It's not something that natural selection causes. So with natural selection, the change in the gene pool is due to one trait being preferred over another. In genetic drift, it is completely random. So there's no reason that any certain allele is lost or gained. It has nothing to do with how fit the individual is. It's completely random. So it happens all the time, though. So it happens to all populations of organisms. But the problem is it's very impactful on small populations. And the reason for that is small populations are typically less diverse to begin with. So they have smaller numbers of individuals. They have smaller numbers of each type of allele because of that. So you have a higher chance of getting rid of an entire allele. Now, there are two main types of genetic drift. We're going to go through both of them. You have the bottleneck effect, and then you have the founder effect. And they are a little bit similar, but we'll go through them and we'll distinguish them from each other. So, one way that genetic drift can happen is through a bottleneck effect. And a genetic bottleneck occurs when there's a drastic decrease in the population size over a relatively short period. So you'll have a normal semi-stable population, you'll have some oscillations in population, as you always do, but all of a sudden you drastically lose a whole bunch of your population. So you go from having all this diversity up here to possibly losing a ton of diversity down here, and that's completely random as to what you lose. So it's not due to natural selection, like I said, it's just a random chance event that cuts the population. So there's a ton of things that can cause a genetic bottleneck, but I'm just going to use one example. Let's go with overfishing for now. Imagine you had a population and you had 25 fish, right? The fish came in 5 different colors. Now, it's not always about color, but color is the easiest thing to show in a PowerPoint, so we're going to use color. There are a ton of other genes that are important that you could lose, but we are going to talk about color for now because it's easier to show. So you have your 25 fish. They come in 5 colors. You have blue, yellow, red, green, and black. And a boat comes along and scoops up 18 of the fish. And it just so happened that those were the fish that were closer to the surface of the water. It was just a random chance that it caught those. There was no rhyme or reason as to where the fish were swimming. So now, instead of 25 fish, you're left with 7 fish in the population. So look at these 7 fish and take a second. Think about, do the fish represent all of the alleles in the same proportion as the original population? So you'll remember the original population had 5 of each thing, so they were about 5 of each color, so they were about 20% of each type of each color. So each phenotype was 5%, 20%, sorry. So now you have these 7 fish. And instead of having 5 of each fish, now we're down to 3 yellow fish, right? 2 green, 1 blue, 1 black. And we've completely lost the red fish. Now, when the population recovers, we're going to see a different breakdown of the alleles that we did before. So these individuals are going to breed with each other. And let's say the red fish was a recessive trait. So we lost our red fish, and that was a recessive trait. Now we have this. We have a completely different makeup of the population. We're back up to 25 fish, so we have our recovery in population size, but we don't have a recovery in our genetic variation. So now we have, instead of 20% of each color, we have 40% yellow, 20% blue, 28% green, 12% black, and 0% red. So we've completely lost an entire type of fish. Now, when we look at this in conservation genetics, really what we will be talking about here are alleles, is we're losing all these alleles. So think of this as 5 different alleles, and now we have completely lost one. So the genetic variation just isn't there anymore. So this was just an example using a physical characteristic, but it could have been anything. So it also doesn't necessarily have to end up in an allele going extinct like it did with the red fish. So you can take this example. By drastically reducing the population, the frequency of the blue allele to the yellow allele completely switched. So we had the majority of alleles being yellow over here. It was pretty close to even. And then over here now it's obvious that there are more blue than there are yellow. And if this has nothing to do with natural selection, it could be possible that now the blue allele was the one that was less fit, and now you're hurting the population. So really what I want to stress is the bottleneck and any type of genetic drift is completely random chance. It has no reason for the changes that it makes. It's just by chance. So the other type of genetic drift that we're going to talk about is the founder effect. So the founder effect describes what happens if a few individuals leave a population. So here's our original population, and these guys don't get killed off. Just a couple individuals decide to leave. So these guys all decided to leave. And this group created another new population, and this group created another new population. So now we have 3 populations instead of 1. So maybe these guys lived on the mainland, and each of these went over and founded an island population. They lived separately from the mainland guys. What happens is the few individuals that found the new population determine what the future genetic diversity is going to be. So again, this is completely random. It's not like they left there being like, oh, let's get as many genetic diverse individuals to found this new island as possible. They just randomly happen to disperse. And that can happen as a natural event in life, right? Maybe they're ready to disperse, or it can even happen in something that we caused, or it can happen in the weather. For example, if you have a bunch of birds, there can be a storm that pushes some birds onto a new island when they are swimming, or not when they're flying. They end up landing on a new island and colonizing it. So that's happened before. And what the problem is, if you have only a few individuals, the genetic diversity is lower. So your future populations end up being less genetically diverse. So this one, we started with 3 different alleles, and we ended up with only 2. This one we started with 2, we end up with all the same, only 1. Whereas our original population is still pretty diverse. Now that's not saying this is the only way this can happen. When these guys leave, let's say by chance all of the red individuals had left, then the original population again would have lost all of their red individuals, and they would have lost some genetic diversity as well. So it doesn't necessarily mean the original population is fine. It can cause issues there as well. So the founder effect really differs from the bottleneck effect because it's not a reduction in the population size. It's just a splitting of a population to create new populations, which may have reduced genetic diversity. So one very well-known bottleneck that occurred is cheetahs. Cheetahs experienced a genetic bottleneck. It was estimated to be just prior to the last ice age, so like 10 to 12,000 years ago. And at that time, we lost a ton of large vertebrates. A lot of them went extinct, and a lot of them were just reduced severely in their population. The cheetah was thought to have gone down to only 7 individuals. So those 7 individuals are the ancestors of every cheetah left today. And because there were only 7 individuals, they had very limited genetic diversity. So our current cheetahs have approximately 99% of the same genes when comparing each other. So there's really not much genetic diversity in cheetahs at all. and things aren't getting better for them, unfortunately. In the past 100 years, we've lost about 90% of our cheetahs. There's a lot of factors that go into that. This time it's not just the Ice Age. It has to do with a lot of things. A lot of them are human caused. So we have habitat loss, poaching, over-harvesting, climate change. So pretty much anything you can think of, they're getting hurt by it. It's pretty much all our fault for the most part, and that's going to be a common theme in this class, unfortunately. But recent research has also found that cheetahs are starting to starve to death because their habitat is so fragmented, and it's taking them too long to, they're expending too much energy to find suitable patches to hunt in. So fragmentation is a big issue right now with them. Another factor is the wildlife trade. And unfortunately, cheetah cubs are often stolen from their mothers and sold as pets. And that's because cheetahs are actually relatively easy to tame. You can even teach them to walk on a leash, and people want them as pets because of that. But we're taking them from the wild, and it's taking away new generations of cheetah. But another huge impact, and this actually has to do with their bottleneck effect, is disease. So since cheetahs are also genetically similar, they're able to spread disease throughout their populations much more quickly. And because they're all very similar, they're less likely to have any individuals that are immune to any illness that they can get. So that's all of these things combined has reduced their population from about 100,000 individuals about 100 years ago to just over 7,000 individuals today. So they're in a lot of trouble, unfortunately. Another example of a genetic bottleneck that we cause, unfortunately, again, is the northern elephant seal. And these animals were hunted to nearly extinction in the late 1800s. We actually thought they were extinct for a number of years, but we did discover them. We rediscovered them off the coast of a Mexico island called Guadalupe, and when we found them, immediately we started protecting them. We realized there's not many left. We need to protect them. And it turned out there were only somewhere between about 25 to 125 individuals left in the world at that time. So that's a pretty small population size. And it was made even worse by the fact that they have a harem mating system, which is when one male mates with multiple females. So the same male, there were like a couple of males, they were fathering multiple groups of pups, while other males were outcompeted and weren't allowed to breed. So it was just these couple of males that were passing on their genes to the future generations. So these seals, they're very actually particular about where they'll breed. They only like beaches that are surrounded by cliffs, and those types of beaches are pretty scarce. So the males would show up, they'd establish territories, they'd defend those territories, and anyone who wasn't able to get a territory couldn't breed. The females would only go to the ones that had their own territory. And there would actually be fights between the males. They would kill each other, so they were taking out even more of their population while we were trying to protect them. Really, because of that unequal sex ratio, and the next generation was even more genetically diverse. Was even less genetically diverse. So it's all been exacerbated by the fact that they have such a small population size. Luckily, this is actually a success story. With our conservation efforts, we actually have over 100,000 northern elephant seals today. So another plus is we're not really seeing that many fitness consequences from their bottleneck. And we're hypothesizing that the reason for this is because of that mating system that they have. Because they're used to only having the same couple of males breed, they are genetically predisposed to have low genetic diversity. So they've been doing very well. So one success story. So genetic drift isn't the only factor that can alter genetic diversity. Inbreeding, when individuals mate with close relatives who they share genes, is also a really big issue in genetic diversity. And again, it's a larger issue in smaller populations or fragmented populations. So you're going to notice small populations are more vulnerable to pretty much all changes, which makes it more concerning, combined with the fact that we are decreasing populations in so many of the species all over the world. So inbreeding can lead to higher offspring mortality, fewer offspring, reduced fitness, sterility, and even reduced mating success. So it has a lot of issues. And in general, continued inbreeding leads to unhealthy offspring because it leads to higher levels of homozygosity. So if you think of it this way, if you are closely related to someone, so let's say we have a brother and sister, it can be any species, and you mate with them and you have offspring, you're likely to have the same recessive traits ahead of time. So maybe you're both even heterozygous. That's best case scenario. If you were both homozygous for the same trait, then also not good. But let's say you're both heterozygous and you have offspring. So this is the typical Punnett square for determining the frequency that the alleles will be present in offspring. So the father passes on a big A, little a, mother passes on big A, little a. Now, maybe you have something like the trait for sickle cell anemia, where it's fine to be big A, little a, and it's fine to be big A, big A. But once you get to be little a, and you have two recessive alleles for sickle cell anemia, now your child has that as well. So if you're closely related to someone, you're likely to have those same recessive traits, even though they might not be seen in you. You still might have them, and they're just recessive. So if you mate with someone that's close to you genetically, you're more likely to have offspring that are homozygous for that trait. And you're more likely to have offspring who are unhealthy. So this isn't always a bad thing for like the first time. So there are many species that will mate in necessity with closely related individuals. There is a trade-off there. It's not always the worst thing. Sometimes it's needed in order to keep a population going, and it's better than dying off. But continued inbreeding leads to an overall less genetically diverse population, and it leads to the passing on of these types of traits that you don't want to have. So continued inbreeding causes a lot of population problems. So our next topic is outbreeding. And outbreeding can also alter genetic diversity. So outbreeding occurs when some individuals mate with individuals of a different population. And this can be beneficial. So if you think like Florida panthers, their species was declining, and inbreeding was actually depressing their gene pool. It was making it worse. Some individuals from a different population were added to their population, and there was a significant increase in the fitness of the Florida panther. So they were doing much better. Another example is with the Isle Royale wolves. So these wolves arrived on Isle Royale by crossing an ice bridge that formed between the island and the Canadian mainland during 1948. So they've been there for quite a while. And since the initial population, it's varied from around 50 individuals in 1980 to just 2 in 2016 and 2018. So there's been a pretty steep decline in the individuals. So you can see the moose abundance, which is what they eat, has a lot to do with it. They kind of cycle together. When the moose abundance goes down, their population, there's a little bit of a lag, but it goes down as well. Moose abundance goes up, their population goes up as well. So the moose population decreased so much that the population was starting to decrease on the island. And when you have 50 individuals who have been inbreeding over and over again since 1948, you're going to lose a ton of your genetic diversity. And it was actually found that this population of wolves was more inbred than any other population on Earth. So they were very terribly inbred. They were almost all genetically the same. Their pups were being born deformed, and they were having physical issues. And when they had these last two individuals in 2018, they found one more pup that was like very deformed and didn't survive in 2018 as well. But the two adult individuals that they had left, they realized they needed to do something. And there had been petitions for a while to reintroduce wolves into the island and have them breed with the native wolves that were on the island to increase their genetic diversity. And finally, in 2018, it was approved, and they added 19 wolves. So that's this little increase you see here. They added 19 wolves to the island, and they're doing a little bit better. In the past 2 years, they've only lost 2 wolves, so they're still at 17, they're at 19, because I think they lost 2, and they had the original 2 as well. So they're doing a little bit better. They are hoping to have it be a thriving population. This is still ongoing. It's very, very relatively new that we added these individuals. But it's kind of important to keep them there because they're keeping the moose population in check. And they did actually find one individual on the island that they have no idea where it came from. All of the individuals that they sent to the island were tagged. They had collars, they had tags, and they had marked every individual that they thought was there, but another one showed up recently that they had never seen before. So there's speculation that that's one of the original wolves, or it could have also been a wolf that crossed from the mainland during freeze over in 2018. So there was another ice bridge that could have crossed during. So outbreeding isn't always good. So in the majority of scenarios, it is a good thing. It's a good thing for you to have increased fitness, right? I mean, increased genetic variation. And it usually leads to an increase in fitness, but it doesn't always. Sometimes it's harmful. So outbreeding can disrupt local adaptations of populations. So, for example, there's a species of ibex where different populations were genetically programmed to be born at certain times of the year. And it depended on where they were from. And it had to do with weather conditions. So when new genetics were introduced and these populations started outbreeding, the babies were born too early and they weren't able to survive. So it's not always good. It can disrupt natural adaptations. So there's kind of a balance that you want to produce the most fit offspring. The main goal is something called heterosis. And what that is it's the tendency of an offspring to have superior qualities to its parents. So your offspring is more fit than you because you passed on the proper genes. While inbreeding can be necessary at some point, it's not always a bad thing. It can be necessary. It often leads to something called inbreeding depression, which results in a lowered fitness. So you want to have this maximum fitness here, and it's resulting in lowered fitness. Then when you look at outbreeding, it's often, again, a good thing, but it can lead rarely to outbreeding depression, which we just talked about with the ibex population was one example. So it's kind of a balance. You do want some outbreeding, you might need some inbreeding, but you want to keep your genetic fitness and your genetic variation and your fitness as high as possible, and you want to pass on the best fitness to your offspring. So there are quite a few techniques for measuring genetic diversity. So now we're getting into the specifics of what conservation biologists will do with this. So you want to determine qualitative variation among individuals and populations in the types of alleles that are present at a given locus. So you want to measure the genetic diversity among individuals and populations. And there's 6 basic methods for measuring genetic diversity. But because most of these methods are not used in conservation genetics, we're actually only going to go over 2 of them today. The rest are used for genetic diversity, but not in conservation genetics. The basic tool for all of these techniques is called electrophoresis. So electrophoresis starts by placing purified proteins or DNA molecules obtained from different individuals into separate little wells. So you will put them into each of these little wells here on each sample, and they're on a gel like agarose or something like that. There's multiple different types of gels. So they're sitting on a gel. And then you run a strong electric current through the gel. And because each of those molecules that you put down differ in size or charge, they're going to move across the gel from their origin point at different speeds. So the current is going to cause them to move at different speeds, and because of that, they're going to end up in different positions when you're done. So the final position of the molecules can be determined by using like dyes or radioactive probes. And the final position is telling you what the genetic differences are. So there's a lot of different ways that you can measure the genetic differences. There's a lot of different variations to electrophoresis, but this is the general idea behind electrophoresis. Now in protein electrophoresis, this is your gel, right? So they would have started in these wells here, and however far they traveled, that would be based on their genetic differences. But in protein electrophoresis, the tissues are collected from many individuals, as per usual. They're macerated and they're separated using electrophoresis, and then they're stained to determine the specific enzymatic or protein activity. So comparisons of electrophoresis patterns are made among many individuals, and the gels are scored based on the presence or absence of a particular band. So they'll know where the band that they're looking for is, and if it's there, that will tell them what they're looking at. If it's not there, then they know it's something else. So variation in where the bands are, it can represent functionally similar forms of enzymes or variants of enzymes that are different allelic forms of the same gene locus. And the detection of alleles permits the study of variation among populations or similar taxonomic forms. So this is really how we tell the difference between similar forms. Like you may think, are these 2 things the same species? And you can test it here by seeing, are they going to be exactly the same, or are they different enough to be considered a different species? So we can pull different sections of different genes and test them in here with their DNA. So, for example, if you had 2 alleles, so you had a fast allele, you had a slow allele, some individuals can be homozygous for the fast allele. So that would be like this one right here, which only has the bar where the fast allele is. So that, picture this as if it was like your gel and it traveled up there because it was faster. Some are homozygous for the slow allele. So this one right here, right, only has a bar for the slow allele. And some can be heterozygous. So they might have 2 bars. So it has both of these alleles. So based on where the bar is, how many bars there are, we can determine more about the makeup of the genetics of whatever we were testing. So Then we have microsatellite polymorphism. And in this, DNA banding patterns of individuals at a single locus are visualized using electrophoresis, but this is after we use the polymerase chain reaction, which is PCR. And you don't need to know, if you don't know about PCR, you don't need to go and Google it or know anything really about it. You just need to know that this type of analysis uses PCR first. And what this does is it makes use of the microsatellites, or a specific short sequence of nucleotides that are repeated over and over again. So GA will be your microsatellite repeated over and over again, right? This is the microsatellite, and this is your nucleotide sequence repeated. So with microsatellites, a particular repeat represents a locus. So this is your locus, and the alleles are represented by the number of times that each sequence is repeated. So these will be 3 different alleles, even though they're all repeats of GA, there are different numbers. So that would make them 3 different alleles. So scientists can identify the region that they are looking at on the they can identify the microsatellite by looking for a primer or a flanking region. And those are, they're the same thing. They don't change a very lot. So let's say you know that this allele is surrounded by one specific region of 4 different letters on each side. You can look for those exact same 4 letters in the same order in another individual to find this allele. So typically that's one way to do it. So different banding patterns here, because we're using electrophoresis again, result from the number of simple nucleotide repeats that an individual has. So for an example, if we had AT, 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 so it's 4 repeats, it would produce a different position on the gel than if we had, let's say, 6 repeats. So this technique is really useful for differentiating between really closely related individuals. So it's actually the most common popular technique for conservation genetics. So using this, we can also tell the difference between a heterozygous individual and a homozygous individual. So each line here represents the locus, and you can count the number of repeats to determine the allele. So 1, 2, 3, 4, 5. For example, this first one here has 2 loci with 5 alleles in each. So because there's the same amount of alleles, it makes it homozygous because it's the same. The second example has 7 and 8 alleles, so it's heterozygous because it is different. So microsatellites are good markers because they're widely distributed across the genome, so they're found everywhere. They're highly polymorphic due to slippage during replication. So they come in many different forms, and they're locus specific. So each microsatellite is preceded by and succeeded by a specific primer, which is what I was mentioning before. And that allows us to find the microsatellites easier in different individuals. So we know what we're looking for. We know how to find it. They're codominant, which means they allow us to distinguish between heterozygous and homozygotes. PCR can be used to analyze each of the little SNPs, which is really inexpensive. So it's a cost-effective way to look at this. And then they can also be used at a range of scales. So you can look at it from the individual ID to fine scale phylogenetics, so on a larger scale. So now there are also ways to quantify genetic data. So a couple of terms that we're going to go over. The first one is polymorphism. And what that technically means is the proportion or percentage of genes that are polymorphic. And I know it uses the definition it uses the term in the definition, but what polymorphic is just the frequency of the most common allele is less than 95%. So an example of this, imagine you had a bison population and you randomly picked out 5 individuals. And you looked at the genes of these 5 individuals and you looked at maybe 24 different genes. You found that 23 of the genes were monomorphic. And what that means is that the most common allele found in each of those genes made up more than 95% of the alleles total found for that gene across all 5 individuals. So it was the most common by a lot. But then you had maybe one polymorphic gene. And a polymorphic gene is going to have less than 95%. The most common allele is going to make up less than 95%. So you have your 5 bison here, right? The genotypes can have X or Y. So you can have XY, you can have YY, you can have XX. And if you have XY, that means you have 1 copy of X, 1 copy of Y. If you have YY, that means you have 2 copies of Y. XX means 2 copies of X, right? So to get the frequency, you would just add up all of the copies of each type. So we have 4 copies of X and 6 copies of Y. And then you would divide it by the total number of copies. So 4 divided by 10 is 0.4 and 6 divided by 10 is 0.6. So that makes Y the most common allele there, right? It is there more than the X. But because 0.6 or 60% is less than 95%, that makes this a polymorphic gene, okay? So there's our math. But even though that gene is polymorphic, that doesn't necessarily mean that the whole population is polymorphic. So when you want to find if something is actually polymorphic, which is shown by the letter P, the particular gene might be, but what you have to do is you have to divide the number of polymorphic genes by the total number of genes. And that will tell you what the level of polymorphism is in the individual or in the population. So 1 over 24, because we looked at 24 different genes, is 0.042 or 4.2%. So this bison population, at least based on the 5 individuals we looked at, are only 4.2% polymorphic. So it's a pretty low level of polymorphism. So then we have a similar term, but it has a distinct difference, and that's the heterozygosity. So this is the percentage of genes at which the average individual is heterozygous. It can also be defined as the proportion of individuals in a population that are heterozygous for a particular gene. So it can be looked at multiple levels. We often use heterozygosity as a measure of vigor or health. And when we look at the bison population that we were just looking at, you have 2 out of 5 of them are heterozygous. So what that means is they have an X and a Y, so they're different. These ones down here are homozygous. So to get the heterozygosity, you would just do 2 divided by 5, 2 out of 5. So you would end up with 0.4 or 40%. So that's just for this gene. That is the percentage of heterozygosity for this gene. But again, you can do the same thing you did with polymorphism, and you can get the average heterozygosity for the population. So we have our 0.4, and the formula for this would actually be just adding up all of the polymorphic values that we would have gotten. For this one, because we had 23 that are monomorphic, it would really just be 23 zeros. But let's say we had 22 monomorphic and 2 polymorphic, then we would add 2, we would have 2 values that we'd be adding there. So we'd have the one for this one and then the one for our other polymorphic gene. But again, we have 23 zeros for monomorphic. We have one value for our polymorphic individual, which is 0.4 is our heterozygosity level. So we would add those up, divide by 24, the total number of genes, and we would end up with our level of heterozygosity for the entire population. So that would be 1.7%. Polymorphism and heterozygosity are typically correlated. The polymorphism usually ranges from 0.1 to 0.4, so 10 to 40%, and heterozygosity usually ranges from 0.04 to 0.1, so 4% to 10%. Now, heterozygosity is usually also correlated with population size. So the smaller a population, the less heterozygosity you're going to see. And that kind of fits with the theme of everything we've been seeing today, where small populations really don't do as well as larger populations. So someone actually looked at a bunch of random species from a bunch of taxonomic groups, and they analyzed genetically their polymorphism levels and their heterozygosity levels. And you can see what's interesting is mammals actually have a relatively low polymorphism and heterozygosity, which means they're not very genetically diverse or very genetically different. But I mean, humans, we are a little higher up there. But on average, mammals are pretty low. And what's interesting is the individuals with the highest polymorphism and heterozygosity are invertebrates, because they are a very diverse group of organisms. Mammals are all very similar to one another. So there are some mathematical equations that help you with determining the genetic diversity. And we have our Hardy-Weinberg equations, where they were created by two people, Hardy and Weinberg, and they discovered the idea actually independently from one another, but it ended up being combined because they were so similar. And what they did is they modeled the behavior of alleles in a population. So what they said is that a population's allele and genotype frequencies are constant. They should not be changing unless there's some type of evolutionary force that's acting upon them. So pretty much you would say, like, in a vacuum with no outside forces acting on them, they should be constant. So this was the beginning of population genetics. And it made a few assumptions. So in order to fit under their ideas, you would have to have a large population size. In fact, you'd have to have an infinite population size, random mating, no overlapping generations, which means every individual from one generation has to die off as soon as the next generation starts. No selection, no mutation, and no migration. Now, obviously, all of this is not something that you're going to see in a natural population, right? But this gives us a place to start and an idea of what is ideal. So they had a formula for determining the allele frequencies. So we'll look at our bison example. Now they had big A, little a. We're using X and Y. It's the same thing. We're just using different letters. So it will be the frequency of one allele is P. The frequency of the second allele is Q. So it will be P plus Q equals 1. So that's just saying that the frequency of each allele always has to equal 1 if you add them up for a gene. So for example, we had 0.4 and 0.6. Add them together, you have 1. So it seems kind of simple, but it's just a rule that hadn't been stated before them. So this is just saying it has to always equal 1. So if it doesn't equal 1, it can't, like, it just can't. There would have to, I mean, you can have more than 2 copies here. You can have like multiple other, you can have other alleles, and if you add them together, all of them put together would have to equal 1. So they also had a formula for figuring out the genotype frequency. So what that would be, we discussed genotype, it would be your combinations of alleles. So for the last example, so for X and Y, you can have YY, XY, or XX. So you can be homozygous for either one, or you can be heterozygous. So they used, again, big A and little a for their formulas, but again, it doesn't really matter. So the formula is P squared, which is, for our intents and purposes, we're using Y. So that would be our YY squared. So 0.6 was YY. We would square that, plus 2 times P times Q. So 2 times 0.6 times 0.4, plus Q squared, so that's our Q, 0.4, equals 1. Okay, so when we do all that math, we get 0.36 plus 0.48 plus 0.16 equals 1. And what that's telling us is that 36% of the population is YY, 48% is XY, and 16% is XX. And that equals 100% of the population. So they can also tell us the difference between expected or observed heterozygosity. So because there are factors coming into play here, right? We're not in a vacuum. We do have outside factors happening. We can look at what we see and determine what it should have been without any of these other factors. So often scientists will compare the outside observed heterozygosity with the expected heterozygosity. So we already calculated our observed heterozygosity before, and we had 0.4 divided by 24, and that equaled the 1.7%. And then our expected heterozygosity is 2pq. And that's only when you have 2 alleles. So if you have more than 2 alleles, you cannot use this formula. It is a lot more complicated. But again, this is not a genetics class, so we're not getting into all of that. We're just going to do the simple version right now. So 2pq for this instance would be 0.48. So our expected was 0.48. Our observed was 0.017. So it's much lower than we had expected. We would expect. So there's obviously something else going on there. So heterozygosity can be analyzed within populations. It can be looked at between populations. So either way, but there's actually benefits to each way depending on the type of population you have. And you can actually add together the between and the within numbers to get your total heterozygosity. So total will be HT, and then heterozygosity within is HS, and DST is the difference between total and within, which is actually your heterozygosity between populations. So what that means is within would mean what's your heterozygosity within this one population. Between would be if we average this one and average this one, what's happening between them that's different. So if you have a high DST, so heterozygosity between, then it's necessary to maintain many different populations in order to maintain the species genetic diversity. So alternatively, if you have most of your genetic diversity within every population, then it's less critical to maintain different populations. So this is often actually a really key issue when scientists are trying to manage populations. So we're actually going to get back to this another time when we're talking about populations. But it's important to look at heterozygosity on these multiple levels. So if you look at the example in the photo, each species here, so we'll say green is a species and brown is a species, each species has the same number of big W and little w. but the proportions are different. So within the populations in between. So for example, the species on the left has almost all of its big W's in the population up top, whereas most of the little W's are in the population on the bottom. So what that means is most of their genetic diversity would have to be between the populations. If you lost one of these populations, you would lose the majority of one of these types of alleles. So in this type of population or this type of species, it would be beneficial to protect all of the populations because you need to have all of this genetic diversity. But in the species on the right, each population has a very similar genetic diversity. So they both have equal numbers of big W and little w. So in these types of species, it's not super important to protect the entire species. You can protect one population and you will still maintain the same genetic diversity that you would have had if you protected all of the populations. So it's really a trade-off, and it's a big decision when scientists are trying to decide how to protect a species and how to conserve their genetic diversity the most. So again, population size in this is important. So the larger the population, the larger the heterozygosity level. So you can even see here, so we looked at 89 species of birds, and the heterozygosity level is lower the smaller the population is. So there is a trend here. Now, that's not saying every individual is the same. For example, we have this huge population here with almost no heterozygosity. But on average, there is a trend between population size and heterozygosity. So with the red-cockaded woodpecker, another example, they have pretty normal overall levels of genetic diversity. And that's if you look at the entire species. But most of their variation is between their populations. And this is not a good thing for them, because in order to maintain their heterozygosity, we would need to maintain all of their subpopulations, not just one. So when you do the math, their total heterozygosity is 0.078. Their heterozygosity within populations is 0.011, and between is 0.067. So 86% of their heterozygosity is between different populations. And with this species, it's even more of a problem because when you look at their range map, their populations are very separated from one another. They're not very close to one another. So we would have to protect all of these different areas in order to protect this species. So it's a very hard job being a conservation biologist and determining how to protect the most for the longest amount of time with the resources that you have. This is a big job for something like this. So I mentioned population size comes into play, as it always does. Small populations are more vulnerable to pretty much everything. But when we talk about population size, we're usually talking about the actual population size or the census population size. This is the number of individuals there actually are, okay? But that's not the number that really matters. So you can have a huge population, but what matters is your effective population size. And what that is how many individuals are contributing genes to the next generation. So this is what counts for evolution and for conservation. And it's always less than the census population. It's always less than what we actually have. The effective population, for a simplified example, would be, let's say you have 100 bison. 25 of those bison are juveniles. They're too young to breed. 15 are adults above the breeding age and they're infertile. They're not able to breed anymore. Then you have 60 potentially breeding adults. your effective population size is 60 because these are the only individuals that can be contributing to the future generations right now. So you don't have 100 bison in for your effective population size. You only have 60. And that's really the number that matters. If you could have a huge population of individuals and then have a ton of those individuals being old and unable to breed. Those individuals really don't matter for conservation or evolution because they are past the point where they are able to pass on their genes to future generations. So there are multiple factors that can affect your effective population size. So it's not just that simple idea that I just gave you. One thing that can affect it is the variation in your population size over time. So an extreme example of variation in population size would be, like we discussed, the bottleneck effect, right? You're having an extreme variation in your population size. You can also have unequal sex ratios that will affect your effective population. And we're going to get into those in a little bit more detail. These are the last things we're going to go over today. So populations fluctuate over time. It happens. It's natural. It happens to every population out there. But you need to be able to calculate the effective population size in a population that's fluctuating, because that's the realistic way to look at things. The problem with this is that by calculating your effective population size in a fluctuating population, it's going to give more weight to the small populations, because these are really the ones that are determining what's going to happen for future generations. So, for example, let's say you had a population of individuals. You measured them over 3 generations. Now, the first generation, you had 1,000 individuals. The second one, you had 10. And the third one, you had another 1,000. So that 10 is going to carry more weight than these 1,000 individuals, because that's really a deciding factor in the effective population. Because the next generation, you're going to have 1,000, you got to expect, why did it go down to 10? Might it go down to 10 again? Are there other issues going on? Do they have decreased genetic diversity because we decreased so strongly? So that 10 is really the most important thing we're looking at there. That's why it's going to carry more weight. Oh my God, he's so loud. Hold on. Buddy, stop. Okay. So there is a mathematical way to look at this. So your effective population would be 1 over T. T is your time, your number of generations. So here would be 3. And then you're multiplying that by all of the populations over 1 added together. 1 over all the populations added together, sorry. So you have 1 over 1,000 plus 1 over 10 plus 1 over 1,000. And then for this, you would end up with an effective population, or you would end up with 1 over 0.034. Your effective population then would be only 29.4 individuals. So it's much lower than what you would expect. And that's because we went down to those 10 individuals in the middle there. So you can also calculate the effective population size with unequal sex ratios. Now, the perfect example would be 50 females, 50 males, right? We would have equal males, equal females, but that's not always the case. In fact, it's most likely not the case. In most scenarios, it's not the case. So there's a formula for calculating your effective population size with an unequal sex ratio. And that would be you would have 4 times the number of females times the number of males divided by the number of females plus the number of males. So if we had 96 females and 4 males, which would be a sort of like with the elephant seals, like a harem mating system. So we have 96 females, 4 males that are breeding with each other. We would have 4 times 96 times 4, so there's your female number and your male number, over 96 plus 4. So it will be 1536 over 100, and that equals 15.4. So that's, again, pretty low effective population size when there's 100 individuals there. And these would be the numbers that you would choose after you did the original effective population. So there could have been 200 individuals there, but we're only counting the breeding adults. So that's the same thing with our variation in population sizes. Like, we're only going to be including breeding adults. So that is it for this lecture. I know it was a lot of math, a lot of technical terms. I feel like this is probably the hardest lecture of the class. So if you need help, feel free to email me. Again, I'm going to be putting up the study guide for the midterm. So any math equations that you do need to know, I will put them on there if you need to know them. And I am more than happy to help you guys with whatever you need.